Good afternoon. I'm Ting Leong Chiu. I run the Advanced Imaging Center at the HHMI Janelia Research Campus here in Virginia in America. Joining me are also several people on my team, as you can probably see in one of the windows. Maybe just go around the table there, just so everybody know who you are. 15 seconds introduction is good enough.
Okay, I am Jesse Aaron, and I am the manager of the AIC.
I'm Maya Altaf, and I'm the fellow in the AIC.
I'm Hari. I'm a fellow at the AIC.
I'm Chad. I'm an application scientist.
And I'm Rachel. I'm a data scientist here at the AIC.
Thank you, everyone. Before I go into sharing with you a little bit about how to write a successful proposal, I want to make sure that everybody feel comfortable enough to interrupt me at any moment. So as I put in the chat boxes, the questions that you have in your mind is probably the same questions that is on everyone's mind, or they may not even know that there is a question unless you ask. So feel free to interrupt me at any time. Just unmute yourself. It is always nice to hear questions directly from the attendees. But if you feel more comfortable, you can also type your questions in the chat, and then somebody from the team will read it out and interrupt me during the process. So please keep your questions coming. So what I want to share with you today is basically the keys to writing a good proposal. Writing a good proposal at the end of the day is to get the proposal accepted. That is the main thing. That's the whole point of writing a proposal. So you always have, when you are writing a proposal, you have to think about how the reviewer is going to read your proposal. Don't just keep jamming your science into the paper without thinking about how the reviewers have been asked to review your proposal. Always, when you're writing a grant, when you're writing a proposal, always think from the perspective of the reviewers. Now, if you go to the proposal application website that Michael has very expertly put together, and it is actually quite detailed, and I encourage you to read it very, very carefully, and that is actually the first important thing when you're writing a proposal, is to read the instruction carefully, preferably read it 5 times, and have that with you as you write. Because guess what? The reviewer will be given the exact same instruction. So we are armed with that instruction on how to score your proposal. So you and I have to think pretty much on the same page. So when you go to that instruction page on the website, you will notice that there are 7 sections. And these 7 sections are pretty much the 7 sections that the reviewers are asked to score. Some of them carry more weight. Some of them is just to help us understand what your project is all about. So these 7 sections, as you can see on your screen, include project summary, background, specific aim and hypothesis, and then your experimental approach and design, followed by measurement and quantifiable outcomes, and then the justification for why you want to use the microscope at the Army Imaging Center, and then how this visit, if your proposal is accepted, will impact on you and your laboratory. So these are the exact 7 points that we will be scoring when we are reading your proposal. So now let's go through them one by one so that you know exactly what a reviewer will be looking for, okay? All right, so let me change this to a pointer. Can everybody see my pointer?
Yes, we can see the red dot.
Okay, good. So the very first 2 sections are kind of lumped together. That is just to make it easy for the reviewer to understand what this project is all about. The first one is project summary. This is just equivalent to an abstract that most of you probably have written when you are preparing a poster or even a paper, right? It's about 100 words, very straightforward. Mainly the key point, what are the questions that you want to address, and that's it. And so that in about 30 seconds, the reviewer pretty much know what you are going to be working on. So keep it brief. If you write an abstract more than 100 words, you are doing it wrong. So keep it short. The second thing is the background. Background basically expands the abstract a little bit. And this is a chance for you to frame your questions as to why you want to ask that questions. Usually these are key unanswered questions in your research field, right? And why you think addressing them is scientifically important. So frame that one or two questions. And again, it is a very short proposal. So if you have 5 or 6 questions, you are also doing it wrong. Keep your questions to 2. That's it. And describe the systems that you will use to address this question. You could be using tissue slides coming from patients or tissue slides coming from animal. You could be using a Drosophila. You could be using organ explants. If you are an environmental scientist, you could be, taking water samples from a reservoir, from a river, from a lake, or a soil sample from a farm, from a field. That's all fine, and that get the reviewers into the right frame of mind as they are reading through your proposal, what you're going to be dealing with. Again, keep it very brief, because this too is just to help the reviewer understand your project. It is not going to score you a lot of points. But if you don't do this well, reviewer get confused very quickly, and then no matter how well you write in the subsequent sections, you have already lost the battle. Write it well, but keep it brief. Okay.
If I can,
and then,
can I add one thing, Leon?
yes.
One way that is helpful to keep it brief is just to use like a schematic or a quick figure that overviews, your project, especially if you have a sample that's not commonly used. That can be really helpful to save words and then just show the reviewer exactly what you're working on and keep it quick and to the point. Yeah.
Good. Thank you. Chad is absolutely right. Sometimes a picture is worth 1,000 words. When you only have 200 words to write in the background section, a figure just saves you 1,000 words. And it actually makes it easier for the reviewer to understand what you're trying to do, right? Especially if you're working on an animal, a fish or a bird or something, where the body parts is unfamiliar to the reviewer, a schematic diagrams that you can get from a paper or review articles in the literature, that always helps. Okay.
Leon, I just want to add that, like, for them to keep the questions to maximum of 2 questions, because thinking of the time they're gonna be there, it's like 2 to 3 weeks maximum. So the reviewer doesn't want to know all about your project. It's just like the experiment that you're gonna do at the at AMI and your time that you're going to spend there. So what's the question that you want to address, and what would you like to get out of this 3 weeks only?
Yes, thank you, Mai. Yes, to frame your central questions again, as Mai said, just keep it to 2. Don't give us a laundry list of questions, because that's not going to help. Then this is the 3 sections that will score you the most point, or it will tank your proposal instantly. So it can either work for you or it can work against you. So these are the 3 very important sections, which are the specific aims and hypothesis, your experimental approach and design, and then your quantifiable outcomes and how you're going to do image analysis. So I'm going to spend a lot more time on these 3 sections, and I'm going to break it down into a smaller section. So I want to specifically start talking about how your hypothesis affects your experimental design and your analysis altogether. So this is a busy slide, but focus only on A, the very top row. Focus on A. So usually in your scientific project, what you do is that you have a hypothesis, you plan your experiment, and then you prepare your samples, and then you go to the microscope, you take a few images or a lot of images, and then you start thinking about how you're going to process the image and how you're going to analyze the image. And if everything goes well, you present your data either in a poster or a meeting, or even better, write a paper, right? So this is what we will call a forward flow in experimental execution. The keyword here is execution. This is how you execute an experiment. Now, if you go to the second line, on the other hand, when you think about the experimental planning within this little square, you actually have quite a few other steps. And this is actually how the way you think actually affects the quality of your proposal. So when you are in this experimental planning phase, which is basically the proposal writing phase, you have to think in reverse. Do not think in forward. And what I mean by that is that you got to have the central questions, right? 1 or 2 questions. And then you have to formulate that into a hypothesis. Translate your questions into a hypothesis. Question has a question mark. Hypothesis does not have a question mark. Hypothesis always end with a period, right? And it is a statement that can be challenged by the experiments that you are going to run. So commit to that hypothesis, whether it's right or wrong, doesn't matter. That's what a hypothesis is. That is why you run experiment to test the hypothesis. So make sure that you really commit to that hypothesis. And then based on the way you frame your hypothesis, this now becomes very informative because it actually tells you what kind of results will be meaningful, are needed to test this hypothesis. Without a hypothesis, you are just going to keep doing a lot of experiment and your experiment is not focused. With the hypothesis, it keeps everything in focus. So with the hypothesis, it will dictate the kind of informative results you need. And now that you know what kind of results is informative, it is meaningful for you to test the hypothesis, now you can start thinking one step further, which is what kind of data is required to generate that kind of information, right? So what are the analytical metrics? What are the experimental variables that you will need to measure to generate this informative results to challenge your hypothesis? And with the required data, for example, the cell density, the fluorescent intensity, the speed of something moving, the speed of your cell population dividing, the speed or the proliferative rate or the killing rate of your immune cells in your assays, those are all required data. Knowing all that, the required data, now you can take it another step forward, which is to think about what kind of experimental parameters that are needed for this data. If you are trying to study the proliferative rate, you obviously cannot use a fixed sample, for example. If you want to study something that is differentiating, especially in a whole animal during development, you obviously cannot use monolayer of cell culture. And also, how deep do you need to penetrate into the sample? How fast do you need to be able to follow something that is happening relatively quickly so that you can address the questions? What are the colors that you need? Do you have the right control? What is the color of the control? And how much data do you need to make it quantitative, to make your data analytical? All of this is dictated by the kind of data that is required. So the required data dictates the experimental parameters that is required in your project. With that in mind, you will notice that we finally arrive at the state where we select the right microscope. It is all of these previous steps that determine what microscope you need. Many people that we have seen, not just in Africa, not just in Latin America, not just in Europe, not in America, everywhere. Literally, this is a global phenomena where people start thinking about, I want to use that microscope first, without thinking about all this stuff. So at the end, they choose the wrong microscope, simply because this is a brand new microscope. It is sexy. You have never used it before. You really want to use it. So you are being drawn by the technology rather than, focusing on what is required to address your scientific question. Never let technology lure you into something that you don't need to do. Let your experiment dictate what microscopes you need. And when you get to this point, now all of these, as a scientist, you should be able to do the first 4 steps. This one, if you are not familiar with the microscope, you will probably need help from Karen. You'll probably need help from Michael and all the Army Imaging Center staff. They can help you select the right microscope. But this is very important. All of this will help them guide you to the right microscope. And then you do your experiment several times. And then if you need to redesign the experiment, sometimes it happens, then you can do it over and over again. Okay. So these basically dictates how good your proposal is going to be from a scientific angle. Any questions from anyone or any questions from at the chat room?
Nothing in the chat.
Okay, great, thank you. So your hypothesis. In your introduction or in your abstract, you have written 1 or 2 central questions. And now the very first thing you want to do is you translate that central questions into a hypothetical statement. The keyword here is statement. It is hypothesis does not have a question mark. And phrase your hypothesis in such a way that it can be experimentally challenged, right? And biologists also tend to have this mindset that it is very important for my hypothesis to be correct, because if it is incorrect, then it just proves that I'm wrong, I'm a bad scientist. No, hypothesis to be challenged. Most of the time, hypothesis can be proven wrong. And if you do everything correctly, even when your hypothesis is proven wrong, you learn a lot from it from a scientific perspective. So you should never be protective of your hypothesis. You really challenge the heck out of it, okay? And a focused hypothesis help focus the proposal. So let me give you 2 examples below. Hypothesis 1 state that we hypothesize that chronic inflammation may be involved in driving tissue damage commonly observed in disease X, whatever that is. That sounds like a hypothetical statement. It is in fact a hypothetical statement, but it is a very, very weak hypothetical statement, because this is the word that will kill your proposal, because involve. What does that mean? You actually cannot test if something is involved. So that is a very vague hypothesis that cannot be rigorously challenged by your experiment. So never write vague hypothetical statement like this. If you look at hypothesis 2, it says we hypothesize that disease X leads to an increase in density of CD8 positive cells in the affected tissues, which is pretty much saying the same thing. But now, precisely because it is focused, it is very specific, you can now think about how you can actually test this hypothesis. A control, a normal tissue, and an affected patient sample that has disease X, you can now quantify the cell density of CD8 positive cells in that affected region of the cells, and that immediately test your hypothesis instantly. So 2 hypotheses that pretty much means the same thing. The first one is so vague that it becomes weak and it jeopardized your hypothesis. Whereas the second one is very concrete. It allows you to rigorously test it. So always go for a hypothesis that resembles the second example, okay? Questions? No? Okay.
No questions there, but I will just reiterate that if anyone does have questions, please, you can either talk or send something in the chat.
Leong, I would like to jump in here.
Yes.
Yeah, so just to emphasize what Leong said, in a lot of proposals, people tend to use words like characterize, dynamics, and so on. These are all very vague terms which cannot be measured. So always specify a parameter or a quantitative measure that you're trying to measure.
Thank you, Hari.
We have a raised hand, I see here.
Yes. Go for it.
Yeah, good afternoon. I just wanted to ask on the hypothesis, will you be asking the null hypothesis or the alternate hypothesis?
Can you say that again? I only heard the word alternate hypothesis.
Okay, is it, are we going to, the hypothesis is going to be based on the null hypothesis or the alternate hypothesis?
Oh, this is completely up to you. So your hypothesis has to be usually based on preliminary data, right? you either the preliminary data is generated by you or it is in the literature. You may have read a few papers and say, there are some central questions. And so you can come up with your own hypothesis as long as it addressed your central questions. Or you may say, wow, the whole field is going in this direction. I don't think it is right. I want to suggest an alternative hypothesis. Either way is fine. For as long as it is testable experimentally, it is good. Does that answer your question? Okay, silence.
Yes, thank you.
Yeah.
I'll jump in here too. I really appreciate the participant asking that question, because you're really talking about statistical testing of hypothesis. If you can really design an experiment that will give you significant, and what I mean by significant is statistically significant results, that will be a very strong proposal. So I'm glad that that person had mentioned that.
Yeah. Thank you. Okay. Now, with your hypothesis, you can now set up your specific aims that will basically other what you are going to do when you're at the Army Imaging Center to challenge your hypothesis. So usually one hypothesis can have one or two specific aims. You are also welcome to propose 1 to 2 hypotheses in your proposal. Again, just like mine say, you only have 1 or 2, you can only support, you can only propose 1 or 2 central questions. So if you have 2 central questions, that will translate into 2 hypotheses, right? And each hypothesis can have 1 specific aims or 2. My suggestions is, because this is a very short visit, no more than 4 weeks and in Cape Town, is to not put more than 2 specific aims, okay? So you can have 1 hypothesis, 2 specific aims, or 2 hypotheses, 2 specific aims. They're fine. And in this section, you really want to elaborate the specific experiments that you would like to perform at the Army Imaging Center. And again, it is very important that the experiments are designed in such a way that it can test the hypothesis. And it is also a very common occurrence, even at the Advanced Imaging Center here, when we receive proposal that somebody's specific aim is this way, and then their experiment going that way, the experiments and the hypothesis don't even have any connection whatsoever. That will kill that proposal instantly, because you're doing experiments that has nothing to do with the hypothesis. That's bad. And it is also a good idea sometimes to state what the predicted outcome would be if your hypothesis can be supported. And if your hypothesis is correct, this is what you would expect the outcome to be. And if your hypothesis is wrong, then this outcome is going to happen. So it is good to anticipate a little bit what the outcome would be. And that actually helped the reviewer understand whether or not you have given it enough thought and to know what to do with the data. When the data actually finally come out of the microscopes, you actually know what it means. So having that kind of anticipation, any kind of predicted outcome statement, sends a very strong message that you actually know what the data is going to mean when it come out of the microscope. Okay, and each hypothesis must have its own set of experiments. That goes without saying. And here are 2 examples, again, one good, one bad, obviously, of specific aims. The first one is clearly bad. We will characterize the tissue samples from patient with disease X. As a reviewer, put yourself in the shoes of the reviewer. Do you know what that sentence means? You probably don't. So it just simply means that we're going to look at the tissue samples. That's it. That is what that sentence means. And you cannot score a specific aims like this. That is just bad. And when a reviewer see this, your score starts to go down. Whereas if you look at example 2, it basically says we aim to compare the CD8 positive cell population density in tissues collected from patients and then normal control, right? If our hypothesis was correct, we would expect to see elevated CD8 positive cell density in disease X tissue compared to control. Now, as a reviewer, again, if you are the reviewer, you actually understand what they are trying to do, what they are trying to quantify, and they know exactly what the data actually mean when you acquire it on the microscope. So that is a good, specific, rigorous, focused, specific aim statement. Okay, question? And then your experimental design. This, if you are not sure, the Army Imaging Center people can help you, but I kind of still want to go through this together with you because this is your project. Frankly, nobody knows your project better than you. We can help you with a little bit of technical insights, especially as you get closer and closer and closer to the microscope part of the experiment. But at the end of the day, the design of your experiment is your expertise, and you should not shy away from really telling people what is important to your project. So the detailed design of your experiment should have 5 potential sections. The first one is to provide a brief outline of each step of the experiment. And you describe the experimental conditions and how they will be controlled. For example, like the previous instance, we talk about they're going to compare disease X patients versus control, and then they're going to talk about measuring the CD8 positive cells, for example. Again, we leave it up to you. This is your project. And then you want to describe your labeling approach. What is the antibody that you're going to use? What are the fluorophores that are attached to the antibodies? You want to make sure that you spelled out the fluorophores, because fluorescent molecule has specific wavelength. If you choose a fluorophore that a microscope at the Army Imaging Center does not have the filter for it or the laser to excite it, then we actually cannot entertain that project, even though everything else looked good. And if you may want to use the right dye, and the dye better be able to help you with the experiment. A lot of people tend to just say, well, we got to stain the cell with DAPI. And sometimes DAPI doesn't add anything. And sometimes DAPI add a lot to your information. So don't just simply add a bunch of dyes that has nothing to do with addressing your experiment, okay? Simply because somebody else published it doesn't mean that you have to use it if it doesn't concern your experiment. And it is also very important to state whether or not your sample is going to be live, is going to be fixed, and if you're bringing bacteria or any kind of agricultural samples or yeast or mold or plants or anything, what is the name of the strain of the organism? It is very important, because bear in mind that when you go to South Africa, you are crossing international border, and you're going to be shipping a lot of stuff internationally. So there are a lot of procedures that need to be taken into consideration. Some strain may not be importable into South Africa, or you have to make sure that all those details are very clearly stated. And if you have made any kind of genetic modification into the sample, for example, a HeLa cell or any kind of cells that you have made stable cell line, the plasmid and everything and the virus and everything has to be clearly stated in your proposal for safety reason and for importation reasons. And then you also have to make it very clear what are the imaging parameters. What is the resolution that is required? What is the imaging speed? What is the duration? Speed and duration together, speed and duration is in inverse proportion. If you want to increase the imaging speed, you usually cannot image it for a long duration. If you want to image something for a long time, you obviously cannot image it very, very fast, because your fluorophore is going to bleach. So think about how you're going to balance that. Again, if you're not sure, seek consultation. And the time interval is important, and the volume of your image, how deep and what is the area, so that together prescribe the cubic micron or sometimes even cubic millimeter that you are going to be imaging on the confocal. And the number of samples, as Jesse has mentioned earlier, you need to think about statistics. Now, do not bring in 1,000 slides to the Army Imaging Center. You have 4 weeks. You cannot possibly image thousands and thousands of slides. So think very carefully. And if that scope of your experiment is beyond what is possible, you might want to trim your specific aims down and trim your hypothesis to a manageable level, right? So now let's look at the examples below.
Can I quickly add a couple things on imaging parameters?
Yes.
Sure. So, so two things. One, I just want to reiterate something Leong has already mentioned, is that these are the imaging parameters that are required to test your hypothesis, right? You might want to image really, really fast with, you know, super resolution, but if that's not needed to test your hypothesis, then it's only going to complicate things. So really, really focus on selecting imaging parameters that are actually required to challenge your hypothesis, not the best possible imaging parameters that'll give you the prettiest movie. The other thing I want to mention is that if you put imaging parameters in your proposal, you're not necessarily going to be tied to those when you get to the imaging center. If you say you want to image every, you know, 1 frame every 30 seconds, it's not like, you know, Mike is going to sit there and make sure you image at 1 frame every 30 seconds. It's a rough guide to what your project is. And that's often iterative when you get there. You know, you take some data, you analyze it, you realize you might need to image a little faster. And so the next time you're at the scope, you image faster or slower, you know. So they're a good guide for
maybe
the
also
reviewer.
somebody from the AIC team can also talk a little bit about creating a table, priority, all that stuff. I'll let you guys jump in.
That's hugely helpful for a reviewer. If you can summarize what you want to image, how you're going to label it, and maybe even some of the imaging parameters. Again, Chad is right. You don't have to, we're not going to hold you to the exact seconds that you want to image. But if you can talk about how you want to image it as a good estimate and summarize all of that in a table, that is hugely helpful for a reviewer, because it just, again, a picture is worth 1,000 words.
And order the experiments by priority. Basically, since you have only 4 weeks, you should be able to cover the must-have experiments which will answer your hypothesis. And if you have time, then you may be able to do some additional experiments. But it would be very helpful if you can order all these experiments in a priority table.
Yeah, priority table is important, as Hari said, because there are just to expand on that a little bit. When you go to a place like the Army Imaging Center, there is always a set of experiment that you absolutely need to do, and that is called a must-have experiment. And then you will be hoping to be able to also run a little bit of your dream experiment that, as Michael has challenged you earlier, is things that you want to do that is outside of your comfort zone, that is really pushing the envelope, and that is a nice to have experiment. You can either write 2 proposals, as Michael has suggested, or if you are not brave enough to submit 2 proposals, you can write 1, but you can also include them as like, okay, if I have extra time, I would love to be able to also do some of these experiments. So those are called nice to have experiments. So the must have experiment and the nice to have experiment, you can summarize all of them in a table instead of, you know, writing 5-6 paragraphs of words to describe it, and at the end confuse the reviewer. A table sometimes just send a very clear message, and it saves you a lot of words too. We don't count words in the table, so you're fine. Okay, so good. So now let's look at the examples below. This is actually a very well designed, a very well written experimental design. And it basically samples of diseased intestine will be ethically obtained from the patients, and the sample will be fixed with formaldehyde and embedded, and then sectioned into 50 micron thick slices. Now, straight away, as a reviewer, this is a very important step, because it actually tells the Army Imaging Center whether or not the microscope can actually image something that deep. 50 micron usually is not a problem. If you don't mention this, then the reviewer is left to wonder, how thick is that sample? Is it possible? Anytime the reviewer starts to have doubt or confused, your point starts to drop. So the more specific information you can give the reviewer, the more helpful it will always be, right? And then you can say each slice is processed as standard and labeled with primary goat antibody, anti-CD4, and then secondary rabbit anti-goat antibody conjugated to Cy5, right? So that is the dye. Good. That all makes sense to the reviewer. And then the sample will be mounted on cover slip using glycerol mounting media. That's fine with DAPI. So you're going to stain with DAPI. You're going to stain anti-CD4. So this becomes important because now the reviewer knows that you're going to quantify the total number of cells using DAPI, because DAPI is going to stain all the nuclei in your tissues. And then all of that, you're going to see how many of them are CD4 positive, for example. So that 2-3 sentences sums up your experimental design very well. And then for acquisition, you'll be imaging 40 slices, each of disease and control samples. So a total of 80 slides with 20x magnification, blah, blah, blah. So this is the kind of specific information that a reviewer is looking for. Again, if you are not sure, talk to the Army Imaging Center, okay? Request consultation if you're not sure. This is absolutely critical. If you're not sure about the technical details, request consultation. And then this is also where a lot of biologists don't do very well in their proposal, and that is the quantitative measurements. You have to define the experimental variables to be measured, right? The measured variables, cell density, fluorescent intensity, the particle movement speed, the rapid change of angle of movement, cell death, whatever it is, that measured variables must absolutely challenge the hypothesis. You will notice that everything leads up to the hypothesis. If you have a bad hypothesis, everything falls apart, right? And so avoid very vague words like spatial temporal dynamics, characterize. Words like this does not help the reviewer. You do not need to know how to analyze the image. AMI can help, because if you are not used to using image analysis software, we don't expect you to know how to analyze the image. But as a scientist in your own field, writing a proposal from your own field, you need to know what you need to analyze. The what is what the reviewer will score you. So make sure that you actually spelled out as a scientist what you think you need to analyze to challenge the hypothesis. So here again is an example. The DAPI signal intensity will be used to segment the nuclei so that the computer can see the nucleus, and then which will be used as the marker identifying all the cells, right? And then nuclei that are within a defined distance from the threshold Cy5, this is your CD4 positive signal, will be designated, and then the proportion of CD8 positive cells relative to the total cell number will then be calculated per square millimeter, and then compare between tissue locations, and then also the various disease states. So this is the kind of statement that will be very helpful to show to the reviewer that you know what to do when the data come out of the microscope, okay? Question at this point, anything in the chat room, guys?
There are some questions that Michael have already answered.
Okay. Again, if you're not sure.
So I shine issue abin da ya sa mata only 300 level saboda babu record din ta mu ai an mata changing ID number so aka mata register sabo. So iya wannan level din yanzu mu a record din.
I don't think he's talking to us.
Sorry, Soy, would you mind muting yourself?
Okay, sorry, I wanted to ask a question.
Yes.
Sometimes, like, if you want to use, what do you call it, Drosophila, for example, a model.
Yes.
And do you have to come along with the sample, or you have to get the Drosophila there? And
yeah, I think in that case, yeah, any kind of samples that are live I would assume that you have to come with a sample because I don't think University of Cape Town will be able to supply all those specimens for you. Yes.
Yes, that's correct, Leon.
It's okay. Okay, that answers it.
With samples that are live, generally we'll have to investigate the possibility of getting the live sample to the University of Cape Town. Fortunately, with regards to mice studies and rat studies, there is the potential to use existing colonies that are located on the campus. But obviously all of this is project dependent. So it would be much better to discuss this on a one-on-one. And I recommend that you maybe contact me via email, or you can set up a consultation and we can discuss it.
Thank you, Mike. Good. Any other question? Okay. And then now we're going to the last couple of sections. The first one is justification of need. This one should not be hard for most of you to write. You want to explain why you need the resources at the Army Imaging Center. You can always say that at your local institution, you do not have a confocal microscope, you don't have a slide scanner, but you have a wide field microscope or a simple teaching microscope that cannot do the job, and you absolutely need that technical specs, the ability to resolve something, the ability to be optically sectioning something using a confocal, to be, for example, to be able to image live cells, or to screen a few compounds, that kind of things, that I think most of you can write pretty well in that section. And it is always a good idea to discuss the limitation of microscopy resources at your home institution also. Now, it is actually very good. In fact, even if you have good microscope at your home institution, you can actually include some preliminary data and say, look, I have a wide field microscope in my home institution. I've captured some images. It looks like this. I can see something, but I cannot, it doesn't give me the necessary information to really test my hypothesis because I cannot image it live. I cannot have enough resolution. I don't have the right filter, things like that. That kind of preliminary data, even though they are negative data, help justify why you need to come to the Army Imaging Center. So don't shy away from including negative preliminary data in your justification section. So an example is below.
You have a raised hand. I just want
to. I think Elaine wants to ask a question.
Sure.
Yeah. I have a question about my project.
Yes.
Yeah, I'm working on anti-cancer effect of some natural compound on breast cancer cell line. Mm-hmm. Yeah, on breast cancer cell line.
Yes.
Yeah, so I want to know if in South Africa you have that cancer cell line, breast cancer cell line.
Yeah, I think we can discuss this outside of this workshop, Elaine, because we would need to know exactly what strain you're referring to, but it is possible. So I think if you're able to email me, I'll put my email in the chat and then we can continue this after the workshop.
Thank you,
okay.
Mike.
Okay. It's okay.
Thank you. All right. Good. Now, an example of justification to investigate the hypothesis of disease inflammation. We need to do a large number of samples, and this is impractical without access to a high magnification automated slide scanner, for example, or to measure the surface area of interaction between cellular mitochondria and Plasmodium parasite. We require three-dimensional image data with resolution below 250 nanometer. At our institution, there is currently no microscope capable of achieving this resolution. So a simple statement like this is enough usually to justify why you need to come to the imaging center. Now, impact is something rather unique for this imaging center proposal. You want to articulate how the project will impact on your research direction and your lab. And now, obviously, most people will write, this huge statement about the scientific impact. While that is good, don't make statements that are so big that it's like, wow, being able to image this allow us to cure a disease, right? So the reviewer is not going to believe it. You only have 4 weeks to work on preliminary data, so don't write huge statement like this. We would rather see impact statements that are more specific. How will the exposure to advanced microscope change the outlook, change the direction of how you're going to do your research? And are you collecting data to be used in a manuscript that you're preparing? And how will you translate this knowledge that you learn to your students, to your colleagues back home? And how will access to microscopy like this help change the way you apply for funding back in your home country? And can the Army Imaging Center even help you reconsider, for example, how the research infrastructure back at your home institution should be configured? So these are the kind of impact statement we're more interested in, rather than, wow, I'm going to go there and cure cancer, right? Which is impractical. So these are some key points that I hope are useful to all of you. And I'll be always happy to answer questions, more general questions, specific questions. You should absolutely ask Michael and Karen. But in terms of proposal writing, approaches and stuff like that, the AIC team here at Janelia are always happy to help. And I believe that this is the last slide. So thank you very much. We have time for some questions, I believe. So go ahead and ask away.
Sure, Prof. Thank you, Prof. I want to ask the guys to pay attention to the chat box where Michael have laid some links. One about a paper about experimental design in hypothesis. The second one is about the Full Army visiting research program announcement. And there is one for a calendar link to book a meeting with him, a 45 meeting with him, for those who want to have specific questions to ask to him. So the floor is open. If you have a comment or a question, please feel free to raise your hand and you.
So I know that the whole presentation will be recorded, and I will share the slide with Amy. And then there's also a paper that Michael and I wrote together a few years back about how to formulate a hypothesis-driven quantitative microscopy experiment. I will share that paper. Mike, you probably still have it, so I don't even need to share. You can send that to Amy.
I've sent it to Amy. I will also post it again in the chat if anyone wants to get it there.
Yeah, and it's not a bad, I'm not sure if you have done that, but you can also put it on the application, website.
Yes, that's a good idea. I should do that. I'll do that.
Okay. Questions? Now it's a good time to we have time, so feel free to ask or discuss anything you want to discuss. Don't be shy.
You can either speak with the microphone or type it into the chat, whichever one you're more comfortable with.
Anything?
While we wait, I'll go. While we wait, I'll go through some of the questions that were asked in the chat during your talk, Leon.
Mm-hmm.
The first question, well, I'm going in back reverse order.
That's right.
Let's see, sorry. So Gege asked if it's compulsory that the sample's fixed. The answer to that is no, but it is very project dependent. Some samples we can accommodate live samples, generally anything that's BSL2, and if we can import it into the country, we are able to support that, hopefully. But if a sample can be fixed at home and it doesn't negatively impact the sample acquisitions and the imaging, then I recommend that you do actually ship a fixed sample ahead of time.
So the other thing is, just let me expand that a little bit. You only image live sample if your experimental questions required you to image it live. Imaging live samples add a lot of challenges. The well-being of your sample, whether or not you have genetically encoded fluorescent protein, to really label your cells, you have to learn how to do electroporation, all that stuff. We encourage people to do live cell imaging, but we encourage you to do live cell imaging when it is absolutely required. If you don't require it, if you're just counting cells under various conditions, there is no reason to keep the cell live, because it actually adds a lot of complication.
Perfect.
Okay.
Thanks, Liang.
Keep going.
I quickly, while I'm hovering over this next question, and then I see that there were 2 new questions further down. Clifford asked if the experiment needs antibodies and fluorescence. Now, to everyone, the answer to that is, again, it's project dependent, and it's dependent on what you're trying to observe. If you are looking at a sample where you need to label biological features with a fluorescence probe, and the best way of doing that is with antibodies, then we do need that information. That being said, some of our microscopes can image without fluorescence, so you're not obligated to perform research that uses antibodies and fluorescence. That's just a very common approach.
Several new questions keep popping up. Yeah, so Lillian asked, Saad basically say, if you want cellular uptake of nanoparticles in 3D spheroids, is it possible? I believe it is. You have to consider, so it's not a microscope problem. It's an experimental logistics problem when you're trying to build spheroids. That means that you have to actually send your cells to Cape Town, and you may have to arrive a little bit early to actually build your spheroids, right? So because you obviously cannot ship a spheroid to Cape Town by mail, it's going to completely disassemble very quickly. So it adds a little bit of logistical issues, but I don't think that is a huge problem. But you have to make sure that you justify and write a very good proposal on that, because anything that is difficult to do, we obviously will want to make sure that it is absolutely practical to do it. Good.
Lillian asked a question, Liang, maybe you might be able to answer this better than I can.
Yes, actually, that is a very good question. So Lillian basically said, I want to ask if every experiment must come with a hypothesis. In biological developmental studies, how can one develop a hypothesis? Very good questions. You can absolutely develop a hypothesis in the developmental biology, and we do that all the time here. The question is, should every project come with a hypothesis? No. There are always, so bear in mind that microscope is also an observational tool, and in fact, it is first and foremost an observational tool. If your project is so preliminary that you don't even know what is happening yet, you can absolutely put a statement in your project that say that it is really premature for us to hypothesize at this point, because we actually don't even know what is going on. And we need a little bit of time to get an idea of what is going on before we can formulate a hypothesis. And that is absolutely possible. I want to make it very clear that we do not want to discourage exploratory projects, because a lot of time, good science comes from exploration, right? But I also want to send an even stronger statement. The fact that something is very complex doesn't mean that it cannot, a hypothesis cannot be formulated. I would challenge you to think innovatively, creatively, and rigorously to absolutely see whether or not you can come up with a hypothesis first. If you're not sure, talk to Mike, as you're going to have a lot of talking, Mike. I can almost guarantee you 80-90% of the time, no matter how complicated something is, you can come up with a hypothesis with our help. But there are also times when we really say, wow, this is really premature, but this is pretty exciting stuff. Let's observe without a hypothesis. So the door is not closed, but it is not very wide open for a non-hypothesis project.
Exactly. Thanks.
So I'll just
Leon.
add one more point here that I think is relevant at this stage. At the AAC, a lot of times we have people come to us and they say, you know, there's two competing models that are in the field right now. Some people think this process goes in one way, some people think the process goes in another way. We would like to test to see what is true. That is an equally, I think, valid, it's still a hypothesis, it's just a multiple hypothesis experiment. And if you can clearly say, these are the models, if model A is true, we'll probably see this. If model B is true, we'll see this. That is also a very good way to couch the aims and a hypothesis in a proposal.
Cool. Thanks, Jesse. There was a question that I want to highlight that was from Joshua. Joshua asked if he can use microbial samples that are potentially available at the University of Cape Town, if he's checking the effects and the mechanism of action of herbal extracts. So the reason I'm highlighting this question is I want to talk about when it's necessary to do a microscopy experiment and when it's not necessary. And I think in Joshua's case, it certainly can very well be necessary to perform a microscopy experiment, especially if you're trying to understand a mechanism of action of how a drug might work. So that all speaks to the experimental design. So I'm not going to talk about that. But I do want to point out, because I've had some meetings with various researchers from across the continent about the ideas of doing a microscopy experiment, and then it turns out that microscopy is not the best way to investigate what they're trying to do. And that, if that happens, that is not a failure on your part, that's not a failure on our part. It's just the best way of doing a specific experiment or to reach a conclusion about a hypothesis might not be microscopy. So we will explore what we can, but if it does turn out that microscopy is not useful, I don't want anyone to see that as a failure. I think that that is an equally good outcome because it gets you one step closer to performing that experiment. We are just not able to necessarily help you with that experiment. But then in your case, Joshua, yes, we can talk about biobanks that are available at UCT. And then also we would need to talk about the experimental design to see what information we can extract when trying to determine mechanism of action of various natural compounds.
Great.
I see Aminu asked a question about receiving Plasmodium falciparum, which is obviously malaria. I will be honest and say that right now we don't have an exact mechanism of receiving various pathological agents such as malaria. It is possible, but this will require some logistical planning from our end and working with you. So if this is something that you would like to pursue, I do recommend that you reach out and contact us, and we can start planning that and investigating that as soon as possible. While there is malaria research at the University of Cape Town, there is no malaria research at our building. So something subtle like that might need us to plan a little bit further in advance. Gege asked again about Helicobacter pylori and whether it's available. I'm not sure. We can ask around and see what researchers are using at the University of Cape Town. And if somebody is using H. pylori, we can certainly ask them if we can use it. But other than that, the next best thing would be to ship some frozen samples into South Africa, which might be achievable. Or let me say, it should be achievable.
So no more question in the chat.
Mike, there's a question about, does the team comprise an individual who deal with fluorescent nanoparticles for imaging bacteria from Kitumile magui?
Yes, I see that. Thanks, Maya. I appreciate that. The answer to this question, Kitumile, is maybe not. We don't have that expertise necessarily, but we do have the background that we would be able to support such a project.
Yeah, I don't think that that will be a concern at all. As a microscopist, we deal with samples, almost everything under the sun. That is not a problem.
Yeah, so that shouldn't be an issue at all. I see, Ibrahim, you've asked whether, various people have now asked about cell lines. If I may ask, I am going to write down all of these, but if you could send me an email so that I would be able to respond to you about whether these cell lines are available, and I'll do that. I'll let you know via email.
I think that the questions is now getting into very specific stuff that really require technical consultation. It's sort of beyond what this workshop, writing workshop is all about. This is all technical consultation stuff because some specific samples, all that stuff has nothing to do with proposal writing anymore. Does anybody have any other questions with proposal writing specifically rather than specific element of experiment? If not, we can call it a day.
Yep.
Will there be participation certificate? This is from Kennedy. Unfortunately, as it stands right now, you will not be able to accumulate CPD points because we are not accredited in any way. But remember that the priority behind the African Microscopy Initiative Visiting Researcher Program is, while there will be benefit in that you will be trained, it's not the priority. The priority is to make sure that you are able to generate data that is usable for research publications and grant applications. So that is the goal, and it's not necessarily to prove that anyone has been trained in a specific technique. Obviously, if you're able to publish the results that you generate here, that speaks to your competency in that technique. But we're not accredited in any way to provide CPD points, unfortunately.
So let me also expand this. This is not a training program. You have to think about the Army Imaging Center is your opportunity to access microscopy. So it has nothing specifically to do with whether or not you can get a certificate. You are given the opportunity to use microscopes. You're not there to be trained. The other questions is about budget. So that is the beauty about the Army Imaging Center. The idea is that we will cover the cost of your travel, the cost of your lodging, and the cost of your experiment within reason. So the idea is that the access to the Army Imaging Center is free. I don't think we will cover, sample shipment and stuff like that. But the reasonable expenses is covered by the Army Imaging Program. So you don't that's the whole reason why there is no budget section.
Yeah, can I quickly add to that, Liang?
Yeah.
Liang is 100% correct. We don't need to worry about the budget initially in your proposal. However, this is not, AMI does not fund your research 100%, not even in the vast majority. We are funding your ability to do the microscopy experiment. So that means we will cover within reason various reagents and consumables that are generally usable by more than one visitor. So generally speaking, this means that we are able to pay for secondary antibodies and potentially some form of cDNA or plasmids purchased from around the world, but we will not be able to pay for everything related to the experiment, unfortunately. We wish we could, but we simply don't have the budget. We would not be able to offer what we do if we paid for everyone.
Yeah, so this is not a grant. It is very important to know that this is not a grant. This is just a funded opportunity for you to access microscopy that you otherwise would not have. That is the best way to look at it. We will not transfer money to you so that you can keep this. Again, it's not a funding opportunity. So you don't write your proposal as if it is a 5-year grant. It is just a 4 weeks opportunity for you to use a microscope. That is the most accurate way to think about this proposal.
Yeah, and in the for more medium to long-term future, if you would like to make a proposal for funding to do research, and part of that funding, or excuse me, part of that research will involve microscopy and microscopes that you don't currently have, we can talk about supporting you in that project proposal, or that grant application rather, on a more long-term basis, but there's no guarantee there, unfortunately. But essentially what we can do is that certain aspects of your microscopy project are already covered for and accounted for, and thereby you can get grant money that can cover the rest of your research project.
Mike, there is a question about technical support. From
I see. Is this from the one from Elaine? Is that what you're referring to, Ame? I see Elaine asked a question about technical assistance with regard to cell culture. While we are able to support you and help you with performing cell culture, it won't be our sole responsibility to maintain and get the sample working, which in essence means if you don't have the knowledge and expertise to do your own sample culturing, we won't be able to assist that, because then essentially we would be doing the whole research, and that's not the priority of the African Microscopy Initiative, unfortunately. But I do recommend, if you don't have expertise in something like cell culture, we can always try and arrange a collaboration through the University of Cape Town and not through the African Microscopy Initiative. But that is a discussion that we would have to have away from this workshop.
Mike, there is a question about how far can the program go in research that requires a specific immunostaining, for instance, PPR virus immunohistochemistry.
In theory, we should be able to support that. That there's nothing indicating that we wouldn't be able to work with that. That should be fine.
Okay.
All right.
Good.
One last question that is more broadly applicable to the audience here. Elaine asks if he needs to use phase contrast microscopy in addition to fluorescence microscopy. That is 100% supportable by our microscopes. So yes, if you need phase contrast or DIC and fluorescence, that is certainly very much within the reason of what we can support. And if you'd like more information, you can either read up more information on the website where the list of phase or contrast generating objectives are there. Otherwise, if you're not familiar with the microscopy, you can email me and we can double check that everything will be suitable.
Okay, thank you, Mike. We are 3 minutes above the time. If there is nothing else to ask quickly, we'll let Prof. Leong give the closing remark, and yeah, we are steps for the call.
Thank you, everyone.
Thank you, everyone, for attending, and a huge thank you, please. I'm sure everyone will agree. Thank you to Ame for hosting this, as well as the AIC for dedicating so much of their time to helping us with this writing workshop. It's really impactful, and personally, I'm very appreciative. Thank you, everyone.
Thank you. Bye, guys.
Thank you. Bye.